It is my pleasure to be a part of this session, and I thank the organizers for giving me an opportunity. Uh, so, without a delay, I will go through my topic now. So, today I'll be talking about the bacterium Shigella, its pathogenesis, lab diagnosis, its antimicrobial susceptibility testing, and briefly about the resistance developing in Shigella. Okay, so Shigella is the most important agent of bacillary dysentery. It is a non-motile gram-negative short bacilli. Belongs to the family Enterobacteriales and Tribasteriacea, which is same as E. coli in the genus Shigella. A bit of the history. It was at the end of 19th century. There was an epidemic of bacillary dysentery occurring in Japan, where Kiyoshi Shiga, he was a Japanese microbiologist. He isolated this bacterium, and later, this bacterium was named as Shigella dysenteriae type 1, which was the first organism of the genus Shigella. And following this discovery, few other related organisms were also discovered and were named to honor the lead workers like Flexner, Sohn, and Boyd. And depending upon their names, we are now classifying this Shigella based upon the biochemicals and the serological characters, that is the somatic O antigen, as subgroups A, B, C, and subgroup D. Subgroup A is Shigella dysenteriae with 15 serotypes. Shigella flexneri is in the subgroup B with 8 serotypes. Shigella boydii in subgroup C with 19 serotypes. These three are physiologically similar, and in the subgroup D, we are having Shigella soni with only one serotype, which is antigenically homogeneous. In Shigella flexneri is the one with maximum cases in the developing countries, and said that Shigella soni is in the developed countries, and Shigella dysenteriae type 1 has got the highest mortality rate and has high epidemic potential. In Indian scenario, Shigella flexneri is the highest one, followed by Shigella dysenteriae, and then soni, and least by Shigella boydii. In Kerala scenario, it is Shigella flexneri at the topmost point, then it comes Shigella soni as the second common isolate. Moving on to the pathogenesis of Shigellosis, as already mentioned, this is highly infective with a minimum infective dose as low as 10 to 100 bacilli. The main mode of infection for Shigellosis is by direct contact. That is, as mentioned earlier, it can be from person to person spread via unwashed hands, which is the most common route, frequently occurring in households, childcare centers, or even there are cases developing mainly with the resistant Shigella strains. Sexual contact is one of the mode of infection. Then contaminated food and water, mainly with infected food handlers or preparing food with contaminated water. Also, there are cases reported by swallowing contaminated recreational water, as in swimming pools. Then the transmission can also occur through fomites like toys, diaper pails, or bathroom fixtures, and mechanically by flies. So the main 5 F's for the transmission of Shigella is food, feces, fingers, flies, and fomites. The main pathogenesis is due to the virulence factors of the Shigella. So first and foremost is the invasive property of Shigella, that is by the IPA proteins and type 3 secretion system, and this resembles the entero-invasive E. coli. And the exotoxin is the second virulence factors. In that, Shigella enterotoxin is the one mostly produced by Shigella flexneri 2A type. And the second one is Shiga toxin or the virotoxin, which is mainly produced by Shigella dysenteriae, which causes local vascular damage. And the third type of virulence factor is the lipopolysaccharide of the gram-negative cell wall, which causes intestinal inflammation. So these are the virulence factors of Shigella. So as sir has already shown this picture, so there will be these bacilli will be reaching the M cells of the Peyer's patches and causing inflammatory reaction, resulting in acute colitis, which is a hallmark of shigellosis. And this colonization will result in the necrosis of the epithelium and result in ulcers. So, the colitis and ulceration of the mucosa is the result or results in the bloody or mucoid stool in dysentery. So, there will be ulceration seen in the intestine. Now, moving on to the laboratory diagnosis. So, the laboratory diagnosis for Shigella, we can divide it into 4 different steps. First will be the specimen collection and initial screening, then culture and isolation of the organism, identification by conventional or automated methods, and final confirmation and typing. So, for specimen collection, for a microbiological examination, specimen collection is the most important one. So, we must use proper specimens and in a proper container, and proper timing should be there. So, the fresh stool sample with blood or mucus is the ideal sample for culture and sensitivity for Shigella. Rectal swabs are said to be unsatisfactory if there is a delay in transport or processing the sample, because the Shigella can easily die off in the dry medium. So, if it is in the rectal swabs, the chances of isolating the organism is less. So, it is always better to get a fresh stool sample. And preferably, the sample should be collected before an antibiotic therapy. And while sending the sample, the labeling of the sample specimen is very important, and it should be with a proper requisition form. And the specimen should be transmitted immediately without any delay, that is, not more than 2 hours should be there while transporting. In case there is a delay in transporting the specimen, we must use a transport media like Sachs buffer glycerol saline or Cariblair media, or the sample should be kept at 4 degrees centigrade, that is, should be refrigerated. And even if it under refrigeration, it's better to reach the lab within 48 hours after collection. So, next we do the initial screening, that is preparation of the wet mount of the feces, which will reveal large number of pus cells and RBCs and some macrophages. So, in the wet film, if we in a dysentery stool, we can see plenty of RBCs and pus cells. The second step is culture and isolation. So, for fecal pathogens, we put the samples in enrichment broth and we use selective media, that is mainly to inhibit the commensal bacteria present in the feces. So here for Shigella, the enrichment broth what we use is selenite F broth or gram-negative broth, and selective media are McConkey agar, xylose lysine deoxycholate agar or deoxycholate citrate agar. So while processing in culture, first we plate that into McConkey agar, which is a moderately selective and a highly selective media like XLD or DCA. And we also put the samples in the enrichment medium and we incubate this. The plates are directly incubated overnight at 35 to 37 degrees centigrade for 18 to 24 hours. And the enrichment broth we incubate for around 6 to 8 hours. And then it is plated onto a selective media like XLD or DCA. On incubation, we have the bacterial colonies. On McConkey agar, we get non-lactose fermenting colonies. And for deoxycholate citrate agar, on DCA, we get colorless colonies. And on XLD agar, we get pink colonies without a black center. And that is confirmed by we prepare a culture smear and do gram staining, which will reveal a gram-negative short bacilli. And on performing the motility test, this Shigella is a non-motile bacteria. And for further identification, we can either do a conventional methods by using the biochemical panel, where few biochemical tests are performed, which shows the following results. And in some exceptions are there for sugar fermenters to identify the different species, like for Shigella dysentery is a non-mannitol fermenter. So like that, we can biochemically identify, partially we can identify which is the strain. Also, we can use automated identification system, but it is of less use because it poorly differentiates Shigella with E. coli. As mentioned in the first one, they do belong to the same tribe, so there will be sharing of ribosomal proteins. So it is a bit difficult to identify in the automated identification systems. So the final confirmation of the colonies can be done by slide agglutination using polyvalent or monovalent sera from the colonies on the culture. So for polyvalent antisera, we can identify the genus specific, and the species identification can be done by group specific antisera. So we have separate antiseras available for all the four subgroups. So with each one, we can identify the species of Shigella. So for type group A, if it is positive with group A, it is Shigella dysenteriae. Group B, Shigella flexneri. Group C, Shigella boydii. And group D, it is Shigella soni. Also, now the molecular diagnosis have been advanced. So for that, again, we can send feces, and here the rectal swabs are acceptable, but you need not send them in BTM. You can just send a normal rectal swabs. Even if it is dry, it is okay. We can process it for molecular diagnosis. So for PCR, mainly the target gene is the invasion plasmid antigen, that is IPAH, which is the gold standard molecular marker. Also, there are PCRs for detecting the Shigella enterotoxin 2. The real-time PCR helps in the quantitative detection, and it's widely used for reference or research laboratories, also for outbreak investigations. So the advantage is highly specific and sensitive, and in PCR even we can detect low bacterial loads, and rapid results are obtained compared to the cultures, which will take around 48 hours to get a result. And also, even if the bacteria is not alive, we can detect by PCR. But the limitations is that we cannot perform a antimicrobial susceptibility testing. So by molecular diagnosis, there is other multiplex PCRs or TACMAN arrays which will help in identifying the species. Also, PCR in some case can be used for resistant gene detection. So we can also detect resistance in the strains we have isolated. In our lab currently in GMC Manjeri, we are having only culture and sensitivity and antiseras. We are planning for PCR and to buy PCR kits for identification of Shigella. And also there is TrueNAT and whole genome sequencing, which is mainly for reference laboratories only. So that is about the main laboratory diagnosis. Now, moving on to the antimicrobial susceptibility testing and interpretation. So, for Shigella, the standard which we follow is CLSI. Depending upon that, we are performing the antimicrobial susceptibility testing. The culture is done on the Muller-Hinton agar, and the colony suspension is inoculated, and antibiotic discs are placed. So the antibiotics which are selected for the antimicrobial susceptibility testing for Shigella, that according to CLSI, we have four tiers are there. Tier 1, which is for routine testing and reporting. Tier 2, depending upon the institution specific rules, we can change. And tier 3 is the multi-drug resistant organism you are suspecting. And tier 4 is the reserved antibiotics which we are using. So for, as per CLSI, tier 1 includes for testing, we have ampicillin, ciprofloxacin and levofloxacin, cotrimoxazole, and the third generation cephalosporins like cefotaxime and ceftriaxone. Tier 2 includes azithromycin, and in Tier 4 is our carbapenems like ertapenem, imipenem, meropenem, and uh, tetracycline is included. So these are the antibiotics which we routinely test for antimicrobial susceptibility for both. We can use this for both Salmonella and Shigella. So on antibiotic susceptibility testing, we are measuring the zone of inhibition, as you all know, and we have certain values given in the standard guide as per the standard guidelines: sensitive, intermediate, and resistant. We measure the zone of inhibition in millimeters. So these are the antibiotics which we test in the AST for Shigella, and these are the zone sizes mentioned for identifying whether it is sensitive or intermediate range or in the resistant part. So which are not routinely tested for antibiotics are, as sir already mentioned, ampicillin, chloramphenicol, cotrimoxazole, and tetracycline. They are not used now because it is mostly of the resistance has developed. And also the first and second generation cephalosporins are not used nowadays. So this is the antimicrobial susceptibility testing and interpretation. Now a word about the antimicrobial resistance in Shigella. So nowadays there is a large cases of resistant strains of Shigella is emerging. So as treatment is an essential part in reducing the death rates and prevalence of the disease. But the antimicrobial resistance have been a major issue in Shigella since 2016, where the MDR strains and even XDR strains are arising. So this antibiotic resistance is carried like any other Enterobacteriaceae family. They are carried on mobile genetic elements which are transferred between the family members of this Enterobacteriaceae, like from any other gram-negative bacilli, by horizontal gene transfer. And there is a dissemination of antimicrobial resistant genes. So there are various mechanisms by which this drug resistance occurs in a bacterium. So as you can see in the Shigella, it starts from the sulfonamides were the first one which was used for the treatment in 1950s, and slowly you can see with the as the years pass, each one drug has been developing resistance. So first sulfonamides was there, and later in late 40s and 50s, sulfonamides developed resistance, and the treatment was switched to tetracyclines and chloramphenicol. Then again, later it developed resistance. So nalidixic acid was used to some part. Again, in 1970s, that also developed a resistance. And recently, even azithromycin resistant Shigella cases have been reported. So there have been emergence of antibiotic resistance of Shigella species all over the world, where Shigella soni ciprofloxacin resistance is there. Even in one of the study from our Govt Medical College Alappuzha, there was a study which shows can see from the 2011 to 2016 the resistance have been increased in almost all of the antibiotics which we usually use for Shigella. So there have been a great emergence of antimicrobial resistance. Even if in our college, the past isolates, Shigella soni was of the greater number isolated, and all the isolates of Shigella isolated for the past 3 years were resistant to ciprofloxacin in our case. Here also, this is the resistance by the National Antimicrobial Resistance Surveillance data, which shows highest resistance was observed in ciprofloxacin and ampicillin for Shigella. Also, there have been an increase in the extensively drug-resistant shigellosis, which as per CDC is defined as the strain which is resistant to all the commonly recommended empiric and alternative antibiotics like azithromycin, ciprofloxacin, ceftriaxone, cotrimoxazole, and ampicillin. So that is an XDR Shigella strain, and by a study, they found that the cases was zero in 2015, but they have increased to 5% in 2022 and to 8.5% in 2023. So there is an increase in the XDR Shigella strains. And The main reason may be because they are suspecting gay or bisexual or other men who have sex with men. MSMs is a case, can be a chances of increase in the antimicrobial resistance. Also, people experiencing homelessness, international travelers, and people living with HIV. These are some factors which may be responsible for the increase in antimicrobial resistance. And in a study from 2011 to 2023 in CDC, they found that the most of the XDR strains were associated with Shigella soni and some with Shigella flexneri. So the problem with the XDR shigellosis is there is no optimal therapy is established for this Shigella strains. So it is better for the clinicians to rely on the AST results from a laboratory to guide for the therapy whenever it is possible. And just as Sir mentioned, we just start the treatment with mild, correct the supportive measures have to be taken, and the sensitivity, depending upon the sensitivity only, we need to start the antibiotics. So that's all my presentation. Thank you.